Kia ora koutou katoa, ko Laurelie Mason aho. Kina mana o tēnei whenua, nā tuahuriri ko tēnei te mihi kia koutou. Ne te mihi mai oha, kia a tātou katoa I hui tahi nei I tēnei rā. I aro hoki ne te nā kaupapa o te rā, tai tonu atu ki te whakatauti. Ko te mano kai miro, nono te ngāhere. Ko te mano kai mataranga, nono te o. Kia ora everybody, my name's Laurelie Mason. I'm the Communications Advisor for the University of Otago here in Christchurch. Warm greetings to everybody. We're a hardy bunch here in Christchurch, and us Cantabs, aren't we? Coming out on a horribly cold night like this. I've been watching that rain radar all day, and it was due to start pouring down just as soon as this meeting started. So thank you all for coming. It's great to see you all here. Going back to that Māori saying of in the mihi whakatau I just told you, let me explain what it meant. The bird who eats only from the miro tree will know only the forest, but the bird who feasts from the pools of knowledge will know the whole environment. And knowledge is what we're all about here, and here today is to hear some, learn some. This is the first of two public talks for 2025 we're giving here on campus. And it's to show off our research and clinical mahi and expertise, and showing what we do here at this campus for the University of Otago, based here in Christchurch. Tonight we're really having a heart-to-heart and getting to the heart of things. Literally, hearing the latest research on the human heart, that incredible muscle inside our chests that beats around 100,000 times a day, 70 beats a minute. We know the sobering stats about heart disease and the causes. It's still a leading cause of death in Aotearoa, 1 in 3, sobering indeed. We know the risk factors. It's the smoking, the high cholesterol, high blood pressure, obesity, diabetes, inactivity, and we know the importance of prevention and early detection. But incredible research and innovation is also delivering answers to unlock the secrets of the heart, how it works and why, ushering in novel treatments and interventions. And researchers at this medical school here, as you will hear tonight, have played and are still very much playing a significant role in this, nationally and internationally. Within our Department of Medicine sits the Christchurch Heart Institute, or CHI. Its researchers are internationally respected, their work groundbreaking, work built upon the pioneering work of doctors and scientists from the early days of this medical school in the 60s, 70s, 80s, 90s. The CHI has around 40 studies on the go at any one time, and it's a true taonga of this university. In the next hour, we're going to hear from 4 of the research team from the CHI, who'll take us through some of the latest promising trials and findings, from world-leading blood test developments to fast-track heart attack diagnosis in the ED, to one trial currently underway, an innovative surgical procedure for atrial fibrillation, new drug trials raising hope for congestive heart failure, to what the future may soon hold with epigenetics and the prospect of tailor-made treatments for heart disease. Let me introduce our first speaker. Associate Professor Phil Adamson is a general and interventional cardiologist with specific expertise in the diagnosis and treatment of coronary and structural heart diseases, as well as a clinical senior lecturer at the medical school here. Phil graduated from the University of Auckland and undertook his cardiology training in Christchurch Hospital. He continued with a further 6 years fellowship training at the Royal Infirmary of Edinburgh, focused on coronary and structural heart interventions. While there, he got his PhD in advanced coronary artery imaging, and since returning to New Zealand, has also completed a Master of Public Health. He remains extensively involved in clinical research with over 100 publications, and is a principal investigator for several large-scale studies funded by the Health Research Council of New Zealand and the New Zealand Heart Foundation. Phil has won a number of awards for conference presentations at European cardiology meetings. His research is cited in multiple clinical guidelines in Europe and the US, research which has substantially changed clinical practice, making cardiac CT the first-line investigation for patients with suspected coronary artery disease. In addition to his part-time private practice, he's a consultant cardiologist at Christchurch Hospital. Welcome, Phil.
Thanks, Lorelei, for a very generous introduction. And thank you to all for putting up with the miserable, gloomy weather out there and making it in for the lectures this evening, which are all fortunately brief. So hopefully they're not. Hopefully they're entertaining. As Lorelei pointed out, I'm Phil Adamson. I'm one of the interventional cardiologists here in Christchurch, and what that means is that I do procedures to open up blocked arteries or replace heart valves or fix other structural problems with people's hearts. And so my talk this evening is around one of the trials of a procedure that we use to reduce the risk of stroke in patients with atrial fibrillation, and in particular a sort of first-in-human device trial of a novel therapy for this. So firstly, what is atrial fibrillation? Atrial fibrillation is the commonest rhythm disturbance that we see in patients who come into hospital. It's particularly common as you get older. Once you're into your 70s or 80s, 5% of or 10% of the population have this. The image on the left here is an animation of a normal heart, and the yellow node at the top of the heart is the sinus node. It's the pacemaker part of the heart, and usually it fires off regularly, and the electrical activity is carried down through the atria, which are the small chambers at the top of the heart, and they squeeze blood into the ventricles, which are the main pumping chambers at the bottom of the heart, and they pump blood to the lungs and to the rest of the body. The image on the right shows a heart that's in atrial fibrillation. And what's happening there is that lots of areas around the surface of the atria are firing off electrical signals which are overwhelming the pacemaker part of the heart, and they make the heart beat rapidly and erratically. And importantly, they mean that the atria don't squeeze in a coordinated manner, and that allows blood to eddy within the atria. Now, the problem with blood eddying is it can clot. And in particular, these clots tend to form in an outpouching that comes off the side of the left atrium that we call the left atrial appendage, or the LAA. Blood clots can form there, and here in the middle is an image of the blood clot as an animation, but also an image from an ultrasound scan of the heart showing the blood clot forming within the appendage. And occasionally those blood clots can leave the appendage and go elsewhere in the body, and in particular they can go to the brain, which is a major complication of atrial fibrillation, where they cause strokes. And so the usual approach that we have to this is to start people on blood thinners, and that reduces that risk. And most of you, or some of you, may be aware of warfarin, which is a drug that we used extensively, routinely for this up until about 10 years ago. More recently, there are some newer anticoagulants, but they work in a similar way. They thin the blood to try and prevent clots forming. Of course, therein lies the inherent problem of blood thinners. So here we have a CT scan of a recent patient in Christchurch, actually, who was on blood thinners for atrial fibrillation, who turned up after a relatively innocuous fall while she was on holiday on a cruise ship. This is her CT scan when she came into hospital at the beginning, but over time she deteriorated, and this is the CT scan later. And what you see is that bright white collection is blood that's accumulated inside her skull. It's pressing on the brain, it's squishing the brain to the other side with disastrous consequences. And unfortunately, that's an inherent problem with blood thinners. So being aware of this, there's been a lot of interest in recent years to try and come up with an alternative way of treating atrial fibrillation, preventing strokes. And basically this comes down to plugs, a variety of plugs that we try and implant within the appendage to seal it off, so that no blood clot that forms there can get out and get into the circulation and cause a stroke. Unfortunately, the atrial appendage is a pretty complex structure, and it varies hugely between people in terms of its size and shape. And we have started doing these procedures within Christchurch. I set up a service a few years ago, and we offer this to patients, but it's not suitable for everyone. The existing devices have some limitations to them, which mean they're not suitable for everybody. This animation sort of highlights the point of what we're trying to achieve, or the problem that we face, which is blood clots forming within the atrial appendage. And we were approached by a company, Origin, who are based in Ireland, And they'd been doing some work to develop a new device that they thought overcame some of the limitations of existing devices, and asked us if we could help them move it from the bench, from their engineering designs, into human patients for the first time. So this is what their device looks like. And essentially it's a plug which is meant to go into that appendage and seal it off so that no blood clots can get out. So they'd already done a lot of preclinical work, a lot of bench testing, engineering, developing a very soft, malleable, woven plug that is unlikely to injure what is a very delicate structure. You don't want to tear it and cause bleeding outside the heart. It's covered with a special membrane which is all designed to help speed up the healing process as the body grows tissue over the outside of it and seals it off. And then what we did is we did some work using CT scans of hearts to print 3D models of real-world patients' atria and their appendages in particular. And we used this to do some bench model testing of devices, implanting them in to see how effectively they occluded the appendage. I'll pass, this is a real-world patient example from Christchurch. I can pass this around. You can have a look at what the appendage looks like and the plug sitting in it. So how the procedure works is we need to get into the left atrium. So that involves coming from the vein at the top of the leg up with a long sharp needle poking across from the right atrium into the left atrium, and putting a catheter, which is a hollow tube, into the appendage. Through this catheter we can push out the device, deploy it within the appendage, check that it's in the right position and that we've occluded the appendage, and then we deploy some anchors, which are tiny barbs basically, which come out from the inside of the device, and they grab a hold of the tissue around the device and anchor the device in position so it doesn't fall out, because obviously that would be a problem. And then we release the device once we're happy with it, and over the next 4 to 6 weeks or so, the body grows tissue over that membrane and seals the appendage off from the rest of the circulation, so no clots can get out into the circulation. So here in Christchurch we did the sort of first in the world human procedures with this device, and I'll just talk you through an example case of one we did. The procedure starts with the CT scan. So we get a CT scan of people's hearts, which gives us very high definition images of the inside of the heart. What I've highlighted here in yellow is the appendage. And what we're trying to understand here is the shape and the size of someone's appendage, so we can plan the size of plug that we need to put in. And we can also do sort of 3D simulated models of what the device will look like, so we can understand how deep into the appendage to deploy the device. These are the actual images from the procedure happening. So these are X-ray images. The patient's asleep on the procedure table at the time. What you'll note is the black structure at the top left of the image. That's an ultrasound probe, what we call a transesophageal echocardiogram probe. It sits in the esophagus or the gullet, and it helps guide the procedure from inside. And the first picture on the left is us injecting dye, contrast dye, which is the black swirling stuff, into the appendage to give us an idea of where the appendage is. And the image on the right is now, once we've deployed the device, we've got the anchors in place, and we're injecting dye again to check that the dye is, that the appendage is occluded. So we're injecting dye beyond the plug and checking that it's occluded. These are the images from the ultrasound probe sitting in the esophagus, showing us on the left the appendage before we've put the device in, and on the right with the device in, and we're seeing that it's compressed and that it's occluding, blocking all of the orifice of that appendage, and nothing's going to be able to get past it. And this is a CT scan from the same patient a few months later, where we're checking to see that this healing process has happened as we expected it to, which is it's got a nice smooth surface on the inside of the left atrium, tissue's grown over it, the appendage is completely occluded, there's no contrast, which is the sort of lighter shade on this, getting into the appendage anymore, so there's nothing that's going to come out of the appendage either. Confirms that we've achieved what we set out to achieve. So we've now completed this first in human study. We did 10 procedures in total. 4 of them happened here in Christchurch, 3 in Auckland, and 3 with colleagues in Melbourne. We've demonstrated that it's feasible to do this, and now we're working with the engineers in Ireland again to make some refinements to the device. There's always things that you can improve about this, but we've got ideas about how to tweak it. but we're also in the advanced planning stages of a much larger trial that will hopefully get underway next year in more centres and more patients. What we need to prove is the long-term outcomes from this. So in conclusion, the Christchurch Heart Institute within the University of Otago, in collaboration with Origin Medical, have successfully demonstrated that left atrial appendage can be achieved with the Zenith device with excellent procedural outcomes and short-term clinical outcomes. And so now larger studies are required and are in process to demonstrate if this is a feasible thing at a large scale for our patients. This is just a photo of some of the team here in Christchurch, the nurses that help us doing the procedures, some of the engineers from the company. I think we're having questions at the end.
Wonderful, thank you, Phil. Thanks very much, Phil. That's great. Next speaker now, Professor Martin Tan is Director of Emergency Medicine Research at Christchurch Hospital and an honorary Professor of Medicine at the University of Otago, Christchurch here. He's one of the most published emergency medicine physicians in Australasia. Martin achieved his medical degree in the UK and completed his emergency medicine specialist training in the UK, Australia and here in New Zealand. Since 2001, he's worked mainly at the ED here at Christchurch Hospital as an emergency medicine specialist. Between 2003 and 2006, he set up and co-directed the University of Otago Christchurch's Centre for Evidence-Based Medicine, and working closely with the CHI research team has contributed to some truly pioneering discoveries, one linked to the timely diagnosis of heart disease or heart attacks for patients coming into the ED. As you will hear, these findings have now been adopted internationally, fast-tracking how heart attacks are diagnosed in many emergency departments around the world. Among Martin's many awards and citations, he was the winner of the inaugural Bevan Medal for Excellence in Health Service Delivery Research. He holds a Health Research Council of New Zealand Fellowship for the investigation of cardiovascular disease in the emergency department. Welcome, Martin.
Thank you very much. So first of all, I have a prop because my co-speaker there was complaining he couldn't tell what the time was. And I may actually have to ask my helpful person in the audience to hold that up for me and press start, please. Otherwise, I'll run way over, I'm sure. So thanks for coming to hear me speak. I'm going to ask the audience a question, but I'm going to come back to it at the end. And if I forget to ask you and give you the reason why, I'm going to you have to ask me as in the question. So are there any lawyers in the audience? No, seriously, hands up. Anybody? Anybody that does finance or accounting? Come on, Harrow, I know you are. Hands up. Anybody that does admin? Okay. Managers? Come on, what does everybody do? You're like all on enigma. Hey, you can't find start, you're quite right. There we are. Thank you. Okay. What I want to focus on later on is the supporting cast, and I'll tell you why that's important. So I'm just going to tell you about a project we've got going on at the moment, but it needs a little bit of context first. Here's some disclosures. So I've split this into two groups. The Christchurch Heart Institute has been incredibly helpful to me. There's lots of other groups that have been working with me, and you'll see actually there's quite a lot of industry there as well. And I put that up partly because it's a disclosure, but partly I'm actually quite proud of the fact that for a lot of the blood tests we work on, industry around the world comes to us first to ask our advice about our R&D programs, because we're actually integral in terms of the science we produce, and I've got a lot of expertise there. And we've actually had the ability to have quite a lot of influence on their big companies like Roche R&D pipelines about what they're going to produce, in the hope that actually be more useful for patients in due course. So patients with heart attack, I really like the Heart Foundation's advert that's came out for the last couple of 2 years that's saying, you know, what does a heart attack look like? And they've got somebody squeezing their chest or jumping up and down or those sort of things. And so there's a guy quietly sitting on a bench at the back who's quietly have a heart attack. I don't know if everybody's seen that, but it's quite a good ad. But I guess the consequence of that is all around the world, there are public awareness campaigns about go to hospital if you have symptoms of chest pain, it could be a heart attack. In fact, now the public awareness campaigns that go to hospital even if you think you might have a heart attack and if you don't have chest pain. And the consequence of this is, of course, what's on the slide above, which is chest pain is around the world globally is number 1 or number 2 most common reason for presenting to the emergency department. Okay, so, and as we'll get to in a moment, emergency departments are quite crowded. So This public awareness campaign, although well-meaning and very important, I'd never not want to have it, does have a consequence that we then have to deal with. And so, although I'd like to be able to talk to you, and I could, in a different lecture, talk about how we've sped people through detection of heart attacks to get them to the cath lab quick or treat them, I'm actually going to talk about the other side of it, because it's also very important, because we don't, as a health system, have unlimited resources. So the impact of having lots and lots of patients come to the emergency department with chest pain is they will contribute, if they're the number one cause, to overcrowding. So overcrowding is an inconvenience, it's not a very pleasant environment to work in, etc. But the main thing about overcrowding is it's very closely linked through a huge amount of evidence to bad outcomes for patients, whether it be delays to antibiotics or delays to pain relief or delays to critical treatment, but it's also related to mortality. Okay, so an overcrowded department is where you are more likely to die. And in this country, in a 2 million patient study, we showed that actually if you come to a department that's overcrowded, you're 10% more likely to be dead in 10 days or in 7 days. Okay, so that's quite a powerful figure. And if that was a drug, it would be a wonder drug that made a 10% difference. So overcrowding is not just an inconvenience. And Phil and I were just looking something up on Google before we started now about juggling. Are there any jugglers in the audience? No. Okay. Well, the question I had was, at what point does it get suddenly difficult? There is an inflection point on juggling, and apparently it's at 6 balls. Okay, you can do 4 or 5, but 6 is really tough. And you go from being able to cope to not being able to cope very quickly in the emergency department environment. So all of a sudden, you drop a ball. Okay, and actually, interestingly, it's not if you have 5 green balls and 1 red one, you never drop the red ball. You drop one of the green ones. One of the green ones will be a little old lady who's got a urinary infection that's quietly dying in the waiting room because no space for her in the rest of the department. Okay, so that's when we're going to get to this about what overcrowding's about, because if you simply don't have enough space or staff to deal with all the patients, you're bound not to get all the things you want done with them in a timely manner. And so that's how it all comes together. And it's not particularly a New Zealand thing, that's a global phenomenon. I guess it's a human exercise, a health services exercise to try and solve that problem. And this is one of our attempts. I'm going to show you in a minute about how we went to go about and do it. So this is what we used to do like 10, 15 years ago. We used to do a blood test, it's called cardiac troponin, but it essentially measures release of muscle from the heart when it gets damaged. And we used to do that blood test on arrival, and then we used to do it about 12 hours later. And we couldn't keep the patient in the emergency department for 12 hours, so we admitted up to the hospital ward, and we do a blood test that night or in the morning. And so you can see every patient on this left-hand side here is being admitted to hospital, which is, of course, very inconvenient, expensive, and burdensome to the hospital system as a whole. We actually worked out that if you admitted one patient with chest pain to hospital, between 20 and 30 individuals were involved in that patient's care during that 24-hour period. Just the duplication is incredible. And if you can actually do it in 2 hours or 3 or 4 hours, you could reduce it to 3 or 4 people only. Okay, so not just the overcrowding the hospital and the department, or just health sources duplication and wastage. Very important. So we set that pathway so we can get rid of what's the green line here. So on the red, and this is why I'm getting back to the public awareness campaigns, we're inviting all these people to come to the hospital, or asking them to, in case they have a heart attack. But only a small red line there has a heart attack, 10 or 15%. In some jurisdictions in the US or Australia, it might even be 5%. So we're admitting all the green patients to the hospital relatively unnecessarily with great burden to the system. So what we wanted to do was get to the right-hand side of the slide and get to a situation where low and intermediate risk patients could be sifted out and discharged without getting to the hospital system. Okay, so that's what we were working on for some time. And this is what happened between well, I'll show you right back actually, but this is what we were able to achieve up to this point. So this is the relentless rise in red of hospital attendances to Christchurch ED. You can see 2 blips, earthquake and COVID. And the yellow line is the relentless rise in hospital admissions as opposed to ED attendances. And this slide here is the number of cardiology admissions we had. Now, Phil admits people for atrial fibrillation as well as other things, but you can see since 2011, that curve has gone down. That's all-cause cardiology admissions, and what we really like to focus on, and for this talk, is the people that have chest pain that don't turn out to have heart attack. We don't really want to admit them to hospital unnecessarily. This is what's happened to those patients. So there's never been a time in 20 years where we've admitted less patients for this problem than we do now. Okay, and this is the way we've done it. So as we go through this next slide now, there'll be down arrows, and that's groundbreaking research being led through the CHI with the help of others and collaborating with others around the world, where we've done research. And each down arrow is research, and each up arrow is us taking the research and driving it into practice and showing the impact of that line. So this is what happened: research, research, implement, and so on. So we've driven that number right down to here. Okay, and the final study, iCare Faster, is one of the things I'm going to tell you about today. Okay, so this is really something we can feel very proud of in New Zealand, because actually there's virtually nowhere else I can think of the world, perhaps, but with the exception of Edinburgh, Phil has also worked, where people have been so effective at reducing admissions safely for patients. So I'm now going to tell you about iCare Faster. One of my colleagues, John Pickering, comes up with the phrases or the acronyms. I can never remember what it stands for, but it sounds good. Because that's what we're trying to do, care faster. So I'm now going to move to what we call point of care or whole blood testing. So these little things you can see here are what we call point of care devices. So they're relatively small, as you can see. There's one held in the hand there, and they've got what we call a fast turnaround time. So you can usually get a result of one of these by the patient within 8 to 15 minutes of the results being needed. Okay, and I'll come to that number again in a minute. They're easy to use, even I can use one. And they come in various sizes and shapes, and they're much smaller than what we normally send to the lab. Now, not only is that machine in the lab big, but you have to transport the sample there, and when it gets there, you have to register it. And when it's registered, you have to put it in a centrifuge, and eventually it will get on a track, and then the machine will run the test. And that actually only takes about 20 minutes, but the result probably won't be back for an hour after you've sent it. So that's actually a problem for reasons I'll explain. And what's changed here is these devices used to have a lower precision than the lab test, okay? But we are the first place in the world to deploy a high precision test, okay? And I'll show you the impact that's had. Okay, so this is the flow of patients. It's a slightly complicated slide, but moving from left across here, we've got a patient arriving, and we've done a history and an examination. And what we'd really like is when we do that history and examination just there, we'd like to have the blood result back when we finished it, because we've gone into the patient cubicle, we've done an assessment, we've made up our mind, okay, well, it depends what that blood test that Poden shows. I'll go and look for it. And unfortunately, it's not there, because we've sent the test to the lab and it's going to take about an hour or more to come back, okay? So we don't sit there at the desk going, I wonder where that troponint's going, I'll give it another tap, see if it's there. No, it's not there, I'll go off and do something else. And actually, you may not get back to that patient for really some time, because when the result comes back, you might be in a trauma with a car accident or dealing with another sick patient. So you can see that all these steps then take place until the doctor actions the result. So it's quite the secondary delay. It's not just the turnaround time of the test, it's actually getting back to the patient and acting on the results. And what we really want to happen is this. We want to do a history and examination, and we want the troponin result to come straight back, and we want to action it and do it bang, 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 okay? And so the key thing is you need the result at the moment you look for it. And so the key thing about point of care is actually 60-minute turnaround for a lab is pretty good, but it might as well be 5 hours if it's not back when I look for it. So that's what you can really achieve. With a point of care test with high precision, I can have the result back 100% of the time. Okay, I can make a decision on every patient every time. I might need to do a second test sometimes, but you can see the power of that. Okay, so. Oh, and there's a couple other things. So we're talking about cubicles being blocked, okay? This guy doesn't look very unwell, okay? But he's come into the hospital with chest pain, and he's been put in a cubicle with a machine that goes ping, okay? We don't have that many cubicles with machines that go ping, but we tend to put the chest pain patients in those cubicles because they need monitoring in case they have a cardiac arrest or an event, okay? And that young guy, who looks really, really well, will continue to block that cubicle for the next 5 hours, while Granny is in the waiting room with her urosepsis trying to get into that cubicle, okay? So what happens now is our nurses get the tracing of the heart, and they get the point of care results 7 minutes later because they've set their smartphones to go back to the machine and get it. And they're actually at the doctor going, excuse me, here's the results. Can I get him out of the cubicle? Okay, that's an unforeseen effect of the study. Okay, so we're now rotating cubicles quickly, and we can get someone else into this cubicle really, really quickly as a result. other things have happened. That when a nurse is looking after a patient for 5 hours because they're having repeated troponins done, they're not with the patient for 5 hours, but there are various direct interventions like the ones I've put up here. Actually, the patient's imprisoned on their bed. They can't get off because they're in cardiac monitoring. So they have to ring a buzzer, someone has to come to take them off the monitoring, they have to walk them to the toilet in case they have a collapse and so on. It takes 12 minutes. So you add up all those numbers and you actually find that there's 21 direct patient minutes spent with a patient. The nurses are very worried about the time it takes to run the test, but it's only 3 minutes. So 18 minutes per patient across 50 patients in our department per day with this problem is 2 nursing shifts saved. So this project was essentially to try and prove that you could improve your clinical decision making, always have the test result back when you wanted it. These are the centres that took part. We finished recruiting now. We recruited 62,000 patients. It's one of the largest studies of type ever conducted in a randomised controlled trial fashion. This has been published. It's our preliminary results in the European Heart Journal. And this is the preliminary results I want to show you. So it's a really weird slide, this, because you can see the zigzag line, which is a gradually increasing median length of stay of patients in our department, and it's going up all around the world gradually. But look at that sudden change. At that very moment, the 1st of August 2023, that instant change on the length of stay, which has been maintained in our department. Okay, so and 30 minutes, which is the median reduction. It may not sound very much, but 22 hours per day is absolutely huge. 8,000 hours a year saved. You can imagine what difference that makes on our ability to move patients through our department. Okay, so probably that's all I want to say. Just to say this is a world first. Now people are learning from us, as I'll show you in a minute. But you know, the CHI led this, and New Zealand led this, and I think we can feel very proud of it as a country. So this is a world map, obviously. I almost managed to leave New Zealand off the bottom, but it's still there. And these are, I'm not going to go through each individual slide here, but all these different colours put together are all the different places in the world that we work with in these projects or help implement similar pathways or publish with or share data with, okay? This is a gigantic sharing operation, and sharing is really important. You can't do this without others, which brings you back to you as the audience and other people in there. So I'm going to pick on Haraway. Harry, just put your hand up. Go on, please. There you are. Haraway's an accountant, is that correct? So I couldn't actually have done any of this without her because she was instrumental to us actually getting some of the grant applications in place, running our budgets, which I'm terrible at. Actually, Chris Charles is in the audience as well. I often go to him and go, Chris, can you help me with this? Because I don't understand any of it, and I'm not going to be able to get the grant application in time if you don't help. Same with legal, same with admin, same with lawyers and management people helping us. It's just, I get a bit of the glory, or the rest of the team does, but that's just a massive effort with all this back staff in there supporting us. So the people the rest of you work in all the other professions that support us that do this, thank you very much. And thank you for listening.
Thank you very much, Martin. And with limited health resources, not just in our country, but all around the world, every country, hospital in the world is grappling with this, getting patients in and out faster, getting them diagnosed with heart attacks sooner. It's not only good for the health service, but obviously for the patient too, that you know whether you've got a heart attack or not. If not, you can go home and, get answers quicker, which is wonderful. Okay, our third speaker now, Dr. Nicola Scott. She's a Senior Research Fellow in the CHI within our Department of Medicine. The focus of her research is based around investigating potential new drug therapies to treat heart failure. She graduated from Otago University with a BSc Honours in Biochemistry and then joined the CHI in 2004 to begin her PhD in the molecular biology of heart disease. On graduating in 2007, Nicola was awarded a Heart Foundation Research Fellowship to continue research in the CHI's Omics Lab. In 2016, she switched her scientific discipline to work in the CHI's preclinical physiology laboratory. She soon took a lead in key studies assessing novel therapeutics for heart failure. She leads numerous projects funded by both the Heart Foundation and the Health Research Council of New Zealand. And last year, Nicola was awarded a Senior Research Fellowship from the Heart Foundation to continue her work and was a guest speaker at the Governor-General's reception for the Heart Foundation last November. Welcome.
Ooh, I've been given a laser pointer. I am a little bit of a loose unit with one, so I'll try and be very, very careful and safe. So, as Laurelie said, I'm a cardiovascular physiologist. And what that means is I'm interested in how your heart and your kidneys and your hormones all work together, either in health or in disease. And tonight I'm going to be talking about heart failure. So, heart failure is a really complex syndrome. It has multiple etiologies. It comes from different sources. It can come on abruptly, it can come on slowly, and that brings with it challenges. When we're talking about our health passion, we like to take any opportunity in the limelight to talk about the really big numbers. And for heart failure, the really big numbers are around about 64 million people worldwide have heart failure. That's probably a bit of an underpinning because we do underdiagnose it. It's the number one cause of hospitalisation for anyone over 65. 1 in 5 of us will have heart failure in our lifetime. And when we have heart failure, it has a profound impact on your survival for the next 5 years. But what does that look like for New Zealand? Currently, we predict about 1 to 3% of adult New Zealanders have heart failure. That's 55,000 people today have heart failure, or the entire population of Rotorua. Heart failure is complex. We don't have a lot of interventions available to us for treating it. So our main mechanism of managing heart failure is through lifestyle interventions and really agile titration of medication. So what does medication look like for these 55,000 New Zealanders? Well, I thought we would liven things up. We're going to play a little game. We're going to play I Take That Bingo. So we're going to have a wee drum roll. And the first thing you might be taking is a diuretic, a furosemide. And this came around about, it's been around for a long time, but this is going to alleviate the congestion in your body. In the mid-80s, we had some really cracking research and we came out with some drugs called the prils, enalapril, captopril, other ones that I can't pronounce. So these are our ACE inhibitors. Followed on from that is a family of drugs that I cannot pronounce, the knowns. These are our mineralocorticoid receptor antagonists. And if your GP or your cardiologist is up with the play and is reading the AHA guidelines, oh, no, we're going to be having some LOLs. We've got some beta blockers. These ones kind of slipped in here, a sympathetic nervous system. So these are probably one of our primary medications. But if you're not taking a LOL, you might have a GP or a cardiologist that prescribes a sartan. So we've got this new range of angiotensin receptor blockers. And we've even got a little purple one up there, a really amazing combination therapy of valsartan and sacubitril. We've got a few new game changers that have come in really recent years. We've got empagliflozin, we've got vericiguat, and we have sildenafil in our armamentarium as well. So there are a lot of drugs up there. There's still a few blank spaces. So you might be adding to this little drug regime with a few other combination therapies that come from the source of your heart failure. But what you take home from this is that's a lot of medications, and we don't always just take one. We take lots of them, and some are good at doing the job that they do, and when they mix together, they're not so good. So how can we improve these medications for people with heart failure? Well, first and foremost, we need to understand what's gone wrong in our bodies. So we have a bit of a complex slide up here looking at some of the hormones that we have. And on the left-hand side, we have the natriuretic peptides, or our heart hormones. And we really, really like these hormones. These are beneficial. They're amazing. They do wonderful things. And on the flip side, on the right-hand side of the slide, we have the renin-angiotensin system. Now, intrinsically, there is nothing good or bad in our bodies. They all have a purpose. But when you have heart failure, this balance gets out of whack. And in particular, our pendulum swings towards the right-hand side. We get an exacerbation of the renin-angiotensin system. And the majority of the drugs that we take, that I showed on the slide before, actually target the renin-angiotensin system. So we're focusing on the right-hand side of the slide. But what about the left-hand side. What about the natriuretic peptides? How can we harness their beneficial potential? How can we augment all of the good things that they do? So let's take a little bit of a closer look at the natriuretic peptides. So what I said was they're heart hormones. So our heart isn't just a pump, it's an endocrine organ. It produces its own hormones. The natriuretic peptides AMP and BMP are released in response to stretch and strain. They go around our bodies, they find their receptors. And on the cell surface, they'll find that little receptor, MPRA, they'll bind and they'll generate an intracellular second messenger called cyclic GMP. And within the cell, cyclic GMP goes on to produce these amazing beneficial actions. Now you might say, well, if you want to get more benefit, why don't we just give more natriuretic peptides? Well, we can, but you do need to have that via an infusion. We also have another mechanism called neprilysin inhibition. So that's an effective treatment as well, but it comes with a few pitfalls. A more selective and specialized way of increasing the benefit from the natriuretic peptides is by harnessing the power of cyclic GMP. And one of the ways that we can do that is by inhibiting our body's natural off switch for the natriuretic peptides. So phosphodiesterase 9 happens to be a little enzyme that sits inside the cell and it breaks up cyclic GMP and stops it from having its biological action. So if we can turn off the off switch, so basically turning cyclic GMP really on, can we get some beneficial actions? Can we get relief of heart failure symptoms? So, phosphodiesterase 9 inhibitors, these are the golden ticket for turning off that enzyme. What do we know about them? Well, fortunately, when phosphodiesterase 9 has been kind of under the radar of a lot of pharmaceutical companies, primarily for potential therapies in Alzheimer's. And in preclinical studies, when they didn't quite get the results that they wanted for cognitive assays, they noted that there were cardiovascular effects. So this is happening in the pharmaceutical world. And independently in the research environment, some really smart research groups were looking at PDE9 and they wanted to know a little bit more about it. First, they looked at where it was expressed, and they noted that PDE9 was expressed in the tissues of the cardiovascular system. It's in the heart, it's in the kidneys, it's in the blood vessels. Those are also the sites of natriuretic peptide action. And then they looked a little bit further and went, well, what happens in health and disease? And they saw that there was a difference in expression between healthy hearts and diseased hearts. So you're getting an indication that this enzyme has a critical role in pathophysiology. And now we've got a compound that inhibits this action. So that's great. We've solved the world's problems. We can just launch into giving it to anyone who needs it. Well, we can't. Oh, there we go. That's where our preclinical studies come in. We are the conduit between the great ideas and the test tubes and the patients in the ward and in the ED. We in our laboratory have a really amazing model of acute decompensated heart failure, and we can scale it from mild to severe. And in this model, we tested PDE9 inhibition. We showed that we had an effective drug. We saw increases in intracellular cyclic GMP, plasma cyclic GMP, and we had so much cyclic GMP we excreted it out in the urine, so we were able to test for that. But at the heart level, we also saw that we were able to improve cardiac function. We could increase cardiac output or pump more blood around the body to alleviate some of those symptoms. We could increase cardiac contractility. We were able to regulate blood pressure, so arterial and venous blood pressure dropped. We also saw that we were able to kind of rebalance our hormones, getting more natriuretic peptides in on the action and reducing the renin-angiotensin system. And when you take these functional changes or structural changes and the hormonal changes, it also contributed to renal changes. So we had increased excretion of salt in urine and salt in fluids. So all of these things are really benefiting the system and alleviating the burden of heart failure. So again, in this beautiful system, we've proven that we have an effective drug. So yeah, we're just going to give it to people. It's going to be amazing. But we can't. We have an evidence-based model here where our clinicians require us to really thoroughly investigate how this medication would work with the other medications that they already give their patients. We can't take away life-saving medication. So our next step is to move into combination studies where we test PDE9 inhibition with all of those drugs that were on the bingo slide. We need to know that they're safe. And at the end of those studies, which we have done, we've come to the conclusion that PDE9 inhibition is great. It's safe, it combines well with other therapies. And what that means for us and for you is that it's really time to go into human clinical trials. And as of this afternoon, Christchurch has been approved to enrol patients in a multinational clinical study of PDE9 therapy. So that's really exciting that we've taken it all the way through from the bench and hopefully by Friday to the bedside. So what I've done over the past 8 slides is take you through my entire lifetime of heart failure therapies. So I'm a 1980s baby. Before I was born, frusemide was our #1 way of alleviating heart failure. We just relieved congestion. Through the 80s and 90s, we had the onslaught of all of the modulation of the renin-angiotensin system, and it has been life-saving and life-changing. In the early 2000s, we've moved into this era of more cardiac targeting of our medications, and it's compounds like vericiguat, which is one of those cyclic GMP stimulators, and our PDE9 inhibitor that are modulating the intracellular environment that is making it more specific to cardiac targeting. The future of heart failure medications, I can't navel gaze, I can't predict, but there are some really exciting trials in the area of microRNAs and stem cells, and even in the avenue of epigenetic modulation. So in the next decade, hopefully we'll have an even bigger slide to share and lots more drugs. And obviously I did not do all of this work myself. I am just presenting the work of many, and these are the main players who I have to thank for this. So thank you.
It was fascinating and wonderful news. Even today, the day of our talk, you heard that good news about those clinical trials. It's wonderful. Well, our final speaker tonight is Associate Professor Anna Pilbrow. She's Deputy Head of our campus's Department of Medicine, as well as being a Research Associate Professor in the CHI, where she leads the Omics Laboratory. Now, I mentioned this before, but I didn't explain what it was. It investigates the influence of genomics and epigenetics in heart disease development and progression, and how genetic variation is linked to actual outcomes for patients here in New Zealand. Anna completed her BSc at Otago, going on to complete her PhD in the field of cardiovascular genetics and genomics. Her research explores how genetic variation contributes to susceptibility for heart disease. She investigates genes and proteins within the heart and blood vessels that are altered in people genetically predisposed to heart disease, and aims to identify new biomarkers that will help identify people at risk of having a heart attack. Anna's working also to identify genetic variants that increase the risk of heart disease in New Zealand families who have a strong family history of premature heart disease. She's been awarded the Sir Charles Hercus Health Research Fellowship, an Otago University Early Career Award for Distinction in Research, a Heart Foundation Senior Fellowship, and in 2021 she was named as the Heart Foundation's inaugural Foundation 100 Fellowship winner. Yeah.
Kia ora Laurelie, and kia ora Phil, Martin and Nick. And kia ora koutou, ko ingarangi te whenua o oku tipuna e noho ana o ki Otautahi ko Anna Pilbrow tōku ingoa. I'm really interested in why heart disease runs in families, and today I'm going to tell you a little bit about DNA and also whether we can use our DNA to predict who might be about to have a heart attack. But before I get underway, I thought I'd share with you the story of Sean Lewis, whose story caught my attention back in 2015 when it was profiled in the Heart Foundation's newsletter. So at the time that this article was written, Sean was a fit 40-year-old non-smoker who had a strong family history of heart disease. And because of his strong family history, he always paid close attention to his heart health and assumed, like most of us, that an absence of symptoms meant he had nothing too much to worry about. But he started to experience pain in his jaw when he cycled to work every day and wisely went off to see his GP. His GP immediately sent him off to have a cardioangiogram, and that angiogram showed that one of Sean's coronary arteries was almost completely blocked. So this is one of those wonderful arteries that supply the heart muscle with oxygen and keep it beating. But lucky for Sean, this blockage was picked up early and he was able to have potentially life-saving quadruple bypass surgery. So even though Sean had a very strong family history of heart disease, needing to have bypass surgery at age 40 still came as a real surprise to him, because he'd always been told by his GP that he had a low risk of a heart attack because of his very healthy lifestyle. So in New Zealand, our cardiovascular risk is typically assessed by our GPs using the New Zealand Cardiovascular Risk Score. And this is a fantastic score that combines a whole lot of lifestyle and clinical information, such as your sex, your age, whether your cholesterol levels and your blood pressure, to predict the likelihood that you'll go on to have a cardiovascular event, such as a heart attack, within the next 5 years. And if we were to calculate Sean's cardiovascular risk score based on the information we know about him in the Heart Foundation newsletter, we would agree with Sean's GP. In fact, we would predict he would have had a less than 1% risk of having a heart attack within the next 5 years. So on one hand, you can see that the cardiovascular risk score is a fantastic tool for GPs to rapidly identify the cardiovascular risk profile of their patients, and so that they can guide their management of those patients' risk factors accordingly. And the GP can be fairly certain that if they see 100 patients just like Sean, only one of them will go on to have a cardiovascular event over the next 5 years. But the problem for the GP and for patients like Sean is that the risk score doesn't tell them exactly which patient that will be. And consequently, all of those 100 patients are treated the same. And this is the reason why our group and many other groups from around the world are really interested to see, can we use information in our DNA to identify those high-risk individuals so that we can target treatment on an individual basis and prevent heart attacks. So in the next couple of slides, I'm going to tell you a little bit about DNA. So our DNA is tightly coiled up in the chromosomes of our cells, and if you were to unravel it, the molecule looks a little bit like a twisted ladder, and those coloured rungs on the ladder represent the DNA code, and certain stretches of that code tell the cells how to make a protein, and those stretches of code are called genes. It may surprise you to learn that all of the DNA sequences of everyone in this room are pretty much identical, but there are certain points along our DNA that we differ, and those small differences are called DNA sequence variants, and that's partly what helps makes us individuals, but it's also what can contribute to our susceptibility to disease. And I can tell you that our susceptibility to heart attacks is around 50% lifestyle and around 50% genetic. But it's been a really amazing time for those of us interested in the genetics of complex diseases. So over the last 20 years, we've had some fantastic advances in technology. When I was a PhD student 20 years ago, it was only possible to look at 1 or 2 DNA sequence variants at a time. Now we can generate data on more than 40 million sequence variants in a single experiment, and that test only costs us about $50 to $100 to run. And what that technology has allowed us to do now is to compare the DNA from tens of thousands of people who have a disease with the DNA of tens of thousands of people who don't have the disease, and we can identify the specific DNA variants which occur more frequently or at a high frequency in the people with the disease than those without. And for the first time, we're now able to identify the exact points in our DNA that influence our inherited susceptibility for a whole range of diseases, including heart disease. And I thought you might be interested to see some of the progress that's been made over the last 20 years. So this slide is a representation of the human chromosomes. And what you're going to see is little dots appear next to specific regions of that DNA, which are pointing to regions that have been associated with inherited risk for a particular disease. And so the heart ones are in red. And if I just flick through this, and you can see from 2005 all the way through to 2014. I'll just stop there because it continues to increase exponentially. You can see the amazing amount of discoveries that have been made over that time. And I can tell you that there are now more than 150 individual points in the DNA that we know contribute to our risk of heart attacks. But the really interesting thing for me is that those 150 points in our DNA are not where we thought they'd be. They're not in our genes. And so if this is a piece of DNA, you can see that our genes are spaced out along the DNA, but there are these long sections of non-coding DNA in between the genes. And 30 years ago, we didn't really know what that non-coding DNA did. And at that time, it was sometimes referred to as junk DNA. But we now know there's no such thing as junk DNA. And in fact, it's what we're discovering is that those long non-coding regions of DNA between the genes seem to be really important for regulating or controlling the way those protein coding genes are turned on and off. And so there's now, and it's in this non-coding DNA that more than 95% of our inherited risk for heart disease lies. And so there's now an international effort to find out exactly how these non-coding bits of our DNA might be so profoundly influencing our susceptibility to heart disease. But can we use this information in our non-coding DNA to predict who might be about to have a heart attack? So each of those individual DNA sequence variants that contribute to your risk only contribute individually a small amount each, but we can sum them all together into something that's called a genetic risk score, and that can give us an estimate of an individual's cumulative genetic risk for a disease. And so on this slide, I'm showing you the distribution of a genetic risk score for coronary artery disease. You can see it's a nice normal distribution, and coronary artery disease is the main cause of heart attacks. And this is data from a large international population study from the UK. And so the people you can see that there are just a few people with very low risk scores down this end. Most people have sort of middling risk scores, and there are a few people with high risk scores up the right hand end. And if we were to draw a line in this blue line here at the point on the score which correlates to a threefold increased risk of having a heart attack, this is now allowing us to identify an additional 8% of people, that's 1 in every 12, who have an increased risk of a heart attack equivalent to, say, having high blood pressure, diabetes, or smoking. And although there's still lots more questions to answer and a number of regulatory and logistical matters to resolve, I can see a time in the next perhaps 10 to 20 years that these genetic risk scores may start entering clinical practice here in New Zealand. But perhaps one of the biggest challenges we face is the fact that most of the data that we have so far is all derived from European populations. So Europeans only represent around 15% of the genetic diversity of all human populations. And so the genetic variants that are important in risk or predicting risk and for heart attacks in Europeans may not be the same genetic variants that predict risk in other population groups. And to illustrate this, I've annotated a world map with some pie charts showing the frequency of one of the top heart disease genetic risk variants. And so the high risk frequency is shown in purple. So you can see that the frequency of the high risk DNA is about 50% in Europeans, the same in South Asians, the same in East Asians, but it's much less in people from Africa and also people in the Americas. And so there is now a really big push internationally to try and identify the regions, the DNA variants that are the most important or most informative for all the populations in the world, not just Europeans, so that we can apply genetic risk scores equitably across populations and so that everyone can benefit. But if there's one message I'd like you to take away from my talk tonight, it's that you're not doomed by your DNA. And this is because it's possible to modify or the chemistry of your DNA without altering the DNA sequence through epigenetic processes such as DNA methylation, where we can get the addition or removal of methyl groups to certain points along the DNA molecule. And this can happen, so this occurs when we have various environmental influences cause the addition of these methyl or removal of these methyl groups. And the methyl groups can change the way those protein coding genes are turned on and off and influence our susceptibility to disease. And these new DNA methylation markers are now emerging as a potential new way that we can monitor somebody's cardiovascular risk through integrating their combined influence of lifestyle, genetic, and environmental factors, and monitor the changes over time. And epigenetics might be one reason why we see this really interesting effect that people who have a high genetic risk seem to get more benefit from making positive lifestyle changes on their cardiovascular risk. So whilst having a healthy diet, exercising and taking the medications prescribed by our doctors is good for everyone, it does appear that people who have the high genetic risk get more bang for buck. And the really exciting thing about this is it shows that even if you do have high genetic risk, you can mitigate this risk at least somewhat through making positive changes. So in summary, the evidence is growing to suggest we can start potentially soon using information in our DNA to improve the way we identify people who are at high risk of a heart attack so that we can develop individualized management strategies and ultimately prevent heart attacks. It looks as if you can modify at least somewhat your genetic risk for heart attacks through having a healthy lifestyle and taking medications. And whilst there are some challenges for us to address in the coming years about how we're going to apply these genetic discoveries equitably, I believe none of these problems are insurmountable, and that it's quite possible we will see these tests enter clinical practice within our lifetimes. Thank you.
Great, thank you, Anna. A great positive note to finish on. And thank you very much. Look, I'd really like to thank our 4 speakers for coming out tonight. They've been absolutely brilliant. I think you'll agree. Phil, Martin, Nicola, Anna, thank you very, very much. Thank you all too for coming here tonight on such a cold night. Now, we've run out of time for questions from the floor, but our 4 experts here will be lingering before they pack up and go home. So if anyone's got any burning questions you'd like to come and put to them, I'm sure they'll give you a couple of minutes of their precious time, and that might be the best way to ask them a question if you need to.